Thanks so much for joining me for the first webinar in our series about the Genes in Space Toolkit. My name is Allison, and I work at miniPCR Bio, one of the founding partners of the Genes in Space contest. Today, we will do a quick overview of genes in space, then we will focus on one method in the Genes in Space Toolkit, PCR. I want to say right up front, this will be an introduction to PCR for students who aren't already familiar with the technique. But along the way, we will highlight genes in space experiments and hopefully get everyone thinking about space biology research. Genes in Space is a free science competition where middle and high school students propose DNA experiments related to space biology. One of the many great things about Genes in Space is that it is an ideas contest. All you need to enter is a great idea. We are entering a new era of space exploration with the potential for long-term missions to places like Mars, but there is still a lot we don't know about the effects of the conditions of space on living things. In terms of the contest, the field is wide open and you can propose to study any topic as long as it involves DNA. Throughout the webinar, we will highlight research questions proposed by past Genes in Space winners to give you a sense of the breadth of the scientific questions you could investigate. Each year, one winning Genes in Space experiment is carried out by astronauts aboard the International Space Station, giving students the opportunity to advance cutting-edge space biology research. Just a few weeks ago, astronaut Dr. Kate Rubins performed the winning experiment from the 2019 Genes in Space contest. Vincent Minucci's experiment aimed to investigate how microgravity impacts the function of the nervous system, and we can't wait to see their results. Applying to Genes in Space is easy. First, you identify an open research question related to space biology. Then you formulate a hypothesis and develop an experimental plan to test your hypothesis that uses one of the methods in the Genes in Space Toolkit to study DNA. The last thing you need to do is justify why your experiment needs to be conducted on the ISS. The application questions are already available on the Genes in Space website, so you can start working on your proposal now. On January 19th, the electronic application form will become available, and you can submit your application anytime before April 12th. This year, the Genes in Space Toolkit has expanded to include 3 ways to carry out a DNA experiment on the ISS. We're doing a whole series of webinars to help you get familiar with the different elements of the Genes in Space Toolkit. Today, we're focusing on PCR. January 27th, you can learn about the BioBits cell-free system, and on February 17th, you can learn about the P51 fluorescent viewer. To tackle PCR, first, we'll talk about what PCR is, then we'll discuss a few of the ways scientists use PCR here on Earth. Finally, we have a special guest, Anna Sophia, to describe her winning Genes in Space experiment that led to the first use of PCR in space. Remember that one of the only requirements for Genes in Space is that you have to propose a DNA experiment. PCR is a way for scientists to make copies of specific regions of DNA. Before we get into PCR, we'll do a quick review of DNA though. DNA contains the instructions for life in all organisms that we know of, from bacteria to plants to animals. DNA is a code composed of 4 letters or bases, A, T, C, and G. These bases are strung together into long strands of DNA, and the important thing is that the order of the letters contains information. I think of DNA as the instruction manual for life. The information encoded in DNA determines how an organism develops and functions. That's why studying DNA in space is so important. Scientists use the term genome to refer to all of an organism's DNA. The human genome is 3.2 billion base pairs long, and even relatively simple organisms like bacteria can have genomes that are millions of base pairs long. Now imagine you want to examine a relatively short DNA sequence, let's say 100 base pairs. Trying to identify just the DNA sequence that you want to study could be like looking for a needle in a haystack, because there's just so much DNA. That's where PCR comes in. PCR allows scientists to take a complex DNA sample, like a person's entire genome, and then make billions of copies of just the DNA that they want to study. To go back to the needle in the haystack analogy, the DNA sequence we care about is like the needle, and it would take a really long time to find it in the haystack of the genome. But PCR finds the needle and copies it over and over again, each time doubling the number of needles by making copies of the copies. By the time PCR is finished, it's like having a pile of needles that dwarfs the haystack. All of this copying happens in a test tube. PCR requires precise control of the temperature of the reaction, so scientists use a machine called a thermocycler to achieve this. This is the miniPCR thermocycler. It's named for the fact that the device is relatively small. You can see that you can hold it in your hand. If you want to learn more about the details of how PCR lets scientists copy specific regions of DNA, miniPCR Bio has excellent resources that are all free, including videos, written explanations, and worksheets. The focus for today isn't how PCR works, but what it allows scientists to accomplish after they have amplified the specific DNA sequence they want to study. The ability to make copies of specific DNA sequences using PCR is vital to almost all types of DNA experiments and analysis. PCR is used to diagnose genetic disease, as well as to identify criminals in forensic investigations with DNA evidence. One of the reasons PCR is so versatile is that there are a lot of different things you can do with DNA that has been amplified by PCR. But it's important to realize that PCR is not used by itself. This is because even after you have billions of copies of the DNA that you want to study, you can't actually see that DNA. Additional lab techniques are used after PCR to visualize, quantify, or analyze the DNA that was copied. Common next steps include gel electrophoresis, a technique that separates DNA by length and allows you to see the size of the DNA fragments, and DNA sequencing, which we'll discuss in more detail now. DNA sequencing is reading the order of the bases, the A's, T's, C's, and G's, in a strand of DNA. There are several different DNA sequencing technologies, but PCR is a required step before most of them for two major reasons. Lots of times, scientists are interested in sequencing a specific region of DNA. So PCR allows scientists to make lots of copies of just the DNA that they care about, then only sequence that DNA, which is a lot easier than sequencing the entire genome. Even when scientists want to sequence the entire genome, PCR is typically necessary because most sequencing methods require a lot of DNA. PCR can be used to copy the DNA so you have enough to sequence. Because DNA carries the instructions for life, knowing the DNA sequence can tell us a lot of information. For example, doctors can diagnose genetic diseases by looking for specific changes or mutations in a person's DNA sequence. For genetic testing, doctors typically start by collecting a blood sample. Our blood cells contain all of our DNA, so PCR is used to make billions of copies of just the region of DNA that the doctors want to examine. For example, if they suspect that a patient has cystic fibrosis, a genetic disease characterized by thick and sticky mucus, PCR is used to copy the gene that causes cystic fibrosis. Once doctors have billions of copies of the patient's cystic fibrosis gene, the DNA can be sequenced. Here we can see a very short DNA sequence from a healthy person without cystic fibrosis, and below, the most common cystic fibrosis mutation. You can see that 3 T's have been deleted. Cystic fibrosis can be caused by several different mutations in the same gene, and knowing the exact genetic cause of a patient's cystic fibrosis can help doctors decide the best way to treat the disease in each patient. As you can imagine, using PCR and DNA sequencing on the ISS opens the door to answering innumerable space biology questions. The team that won the 2018 Genes in Space contest used PCR and DNA sequencing to investigate how DNA is fixed after it gets damaged in space. What space biology question do you want to address that could use PCR and DNA sequencing? I do want to be clear, though, that PCR isn't always followed by sequencing. That was just the example that we delved into today. The key thing I want you to take away about PCR is that it is indispensable for the study of DNA. This is why it's so exciting that we have the ability to perform PCR on the ISS. The machines that scientists use for PCR aren't well suited for use in space, mostly because they're the size of a microwave and can be pretty heavy. It's estimated that it costs around $6,000 per pound to send something just to the ISS, which is actually relatively close to Earth. Especially with long-distance space missions a possibility in the future, it's important to have scientific tools that are small and lightweight. The miniPCR thermocycler fits that bill perfectly, and in 2016 it was used aboard the ISS to perform the first PCR in space. The experiment was designed by a high school student, and we are very lucky that she has joined us today to tell you about her groundbreaking PCR experiment. It's my great pleasure to introduce you to Anna Sophia, who won the first Genes in Space contest in 2015 when she was 16. Anna Sophia has wanted to be an astronaut since she was little, so she jumped on the chance to enter Genes in Space. Anna Sophia was interested in changes to the human immune system in space, and her winning experiment that addressed this question was the first time ever that PCR was performed on the ISS. Today, Anna Sophia is working towards her goal to become an astronaut. She studied molecular biophysics and biochemistry at Yale and is currently enrolled in the joint Harvard-MIT MD-PhD program. We are very lucky to have Anna Sophia join us so she can tell you about her winning Genes in Space experiment.
Hi! I've always wanted to be an astronaut, so I've always been pretty concerned about how astronauts stay healthy. After all, nobody wants a cold all the way to Mars and back. But being in space makes astronauts' immune systems weaker, so it's more likely that they'll end up getting sick. In 2015, I came across a website that asked high school students to design DNA-based experiments to run on the International Space Station. And the first thing I thought of was questions about the immune system. Why do astronauts' immune systems get weaker in space? And then what can we do to fix it? After doing more research, I learned that the immune system has cells called T helper cells that guide its response to pathogens. These T helper cells start out as naive or undifferentiated cells, and then based on signals from the rest of our body, they turn into either Th1, T helper 1, or Th2, T helper 2 cells. Each of these cell types is responsible for a specific kind of immune response that it triggers by producing unique signaling molecules called cytokines. For this immune response to work properly, there has to be a balance between Th1 cells expressing Th1 cytokines and Th2 cells expressing Th2 cytokines. But studies done on space shuttle flights with mice in the early 2000s demonstrated that even really short exposure to microgravity was enough to cause an imbalance in the ratio of Th1 to Th2 cytokine expression. Scientists then thought that the same thing might be happening in humans because they found that astronauts who had just returned from space had a lot more Th2 cytokines in their blood when compared to the blood of people who had stayed back on Earth. Based on this information, I hypothesized that going to space was messing up how the Th1 and Th2 cells were expressing their cytokines. If astronauts' Th1 cells and Th2 cells weren't expressing the right amounts of the right cytokines at the right time, that might explain why our immune response wasn't working properly when we're not on the planet. So for my Genes in Space experiment, I decided to use a special kind of PCR to look at the expression of certain cytokine genes to understand if the T helper cells were expressing the same cytokines in space as they were on Earth. PCR was the ideal way to answer my question about cytokine expression in T helper cells because it allowed me to zoom in on just the genes that code for specific cytokines. You see, I wasn't interested in knowing if the genes were there. After all, almost every cell in our body has the same DNA. Our skin cells, our immune cells, our muscle cells, it's all identical DNA. The reason that different cells function differently is because as cells differentiate, the genes that they don't need are turned off. So even though Th1 and Th2 cells have all the same DNA, so they have all the cytokine genes somewhere in there, Th1 cells will only express Th1 cytokines, and Th2 cells will only express Th2 cytokines. So I didn't want to use PCR to look at the Th cells' genomes. I wanted to look at this difference in expression, and to do that I looked at something called the epigenome. The epigenome is a layer of chemical markers that goes on top of the DNA, like methylation, that controls how it's expressed. See, if your genome were a book, the DNA would be the sentences that you're reading, and the methylation would be like lines of pencil crossing out the sentences that you're not meant to read. Luckily, there's a kind of PCR called bisulfite PCR that lets you see if the DNA sentence that you're looking at is crossed out or not. Now, there's one more important fact that brings my whole theory together, which is in different, totally unrelated cell types, scientists had found that going to space altered methylation profiles. So I'm thinking, what if it's the methylation going wrong and causing these T helper cells to express the wrong cytokines in space? So I propose to look at that. I wanted to take Th1 and Th2 cell samples, keep half on Earth and send half to space, and then once the cells in space had been exposed to microgravity for long enough, I wanted to use bisulfite PCR to see if the genes for Th1 and Th2 specific cytokines were methylated differently, suggesting that they were being expressed differently in microgravity compared to the cells we had kept on Earth. If I saw that the methylation patterns of cytokines in space were different than on Earth, for example, maybe in microgravity, the Th2 cells were acting normally, but the Th1 cells weren't expressing enough of their Th1 specific cytokines, but were suddenly expressing some Th2 specific cytokines, that would help confirm my hypothesis that aberrant methylation profiles were leading to weird levels of cytokine expression in space that might even explain why we're seeing immune system dysfunction in astronauts. Maybe, if we could find a common pattern of space-induced changes in methylation, we could even use similar PCR tests to monitor astronauts' genomes before they started experiencing symptoms of space-induced immunodeficiency. But most of all, I wanted to do this experiment because it seemed like an amazing opportunity to use a cool molecular biology tool to learn more about how our genes work in space.
Anna Sophia's question was about changes in the immune system in space, and there are lots of unanswered questions about the effects of space on our bodies. But one of the most exciting things about space biology research is how many interesting topics there are to study. That's where you come in. We can't wait to read your Genes in Space proposals to see your ideas for groundbreaking space biology research. Now to some of the questions that we received. Do I have to propose a PCR experiment for Genes in Space? This is a great question, and the answer is no. While in the past, Genes in Space experiments were required to use PCR, starting this year, your experiment can utilize any of the 3 Genes in Space Toolkit techniques. A related question, can my proposal use more than one of the Genes in Space Toolkit techniques? Yes, absolutely. For the first time ever, the 2020 Genes in Space finals took place remotely, which raised the question of whether this will also be the case this year. And the answer is yes. The finals for the 2021 contest will be virtual. The presentations from the 2020 finalists to the judges are still available online, and we encourage you to check out these amazing presentations to get more exposure to open questions in space biology research. Do I need a sponsor to apply to Genes in Space? Yes, you do need to list the name of an adult when you submit your application. We encourage you to seek mentorship from a teacher, parent, or another science enthusiast, but your sponsor only needs to agree to have their name listed on your application. Do I need to tell my school I'm applying to Genes in Space? No, although if you're selected as an awardee, your school will receive a DNA discovery system from miniPCR Bio. On to some scientific questions. The contest is called Genes in Space, so this is a great question. What is a gene? A gene is a specific region of DNA that contains the instructions for making a protein. Not all DNA contains the instructions for making protein, but proteins are really important, so lots of DNA research focuses on genes. That obviously brings us to a related question: what is a protein? I think of proteins as the tools for life. They carry out most of the functions in a cell. To give just two concrete examples, the top protein is hemoglobin, which is a protein found in red blood cells responsible for transporting oxygen throughout the body. The Y-shaped protein is an antibody, an important component of the immune system that helps our body recognize and fight off infections. qPCR has been in the news a lot because it's the gold standard for diagnosing infection with SARS-CoV-2, the virus that causes the disease COVID. So what is qPCR? qPCR stands for quantitative PCR, and it's a type of PCR that allows scientists to calculate relative DNA levels. If you want to learn more about qPCR, turn into the Genes in Space Toolkit webinar focused on P51 fluorescent viewer. A related question: can I propose a qPCR experiment for Genes in Space? Absolutely. And one last question: what are some of the benefits of performing PCR in space instead of sending the samples back to Earth? It's not that easy to send samples back to Earth, even from the ISS, and in the future, as we undertake missions to far-off places like Mars, it will be even less feasible to send samples back for analysis, especially when you need a quick answer. For example, does an astronaut have the flu? Is this microbial growth harmless or pathogenic? Dr. Sarah Wallace monitors the microbes growing on the ISS. It used to take weeks to identify samples because they had to wait for them to be shipped back to Earth for analysis. Now, astronauts can collect a sample on the ISS and then use PCR and DNA sequencing to identify the microorganism quickly. We will wrap up with a rundown of some of the logistics for entering Genes in Space. The contest is open to students who live in the U.S. and are in 7th through 12th grade. Students can submit an application on their own or work together in groups of up to 4 students. Applicants must list the name and contact information for a teacher, parent, or other adult to sponsor their application. And the application deadline is April 12th, so there's plenty of time for you to work on a proposal. Finally, I want to emphasize that it is completely free to apply. Again, the contest opens January 19th and closes on April 12th. Awardees, semifinalists, and finalists are announced throughout May. And then finalists are paired with scientists who serve as mentors and help them refine their research proposals. In July, finalists will present to a panel of judges who select the winning experiment to be launched to space. There are tons of free resources available online to help you with your application. Check out genesinspace.org/learn. You'll find videos explaining biotechnology like PCR and DNA sequencing, as well as interviews with scientists conducting space biology research. For example, Dr. Massa describes her research focused on growing plants in space for long-distance missions. What DNA experiments can you think of related to growing food in space? You'll also find Genes in Space application tips, including the detailed scoring criteria. I want to end by reminding you that this year, the Genes in Space Toolkit has expanded. In addition to PCR, which we covered today, you can also propose experiments that use the BioBits cell-free system and the P51 fluorescent viewer. To learn more about these exciting new tools, tune in to our next webinars on January 27th and February 17th. Thank you so much for joining me. We hope to see your application for the 2021 Genes in Space contest.